Okay, the online transmission is already working. In 1 minute, I'll start the recording and we start sharp in 2 more minutes.
I will go to the background. Good luck. Very happy to host this. So good luck.
Thank you, Edith. Okay, well, hello, good morning, good afternoon to all joining this session dedicated to know more about EBRAINS, well, and all the infrastructure dedicated for brain research. My name is Daniel Tamirano. I'm the academic communities manager in Red Clara, and I'll be your host today. Just 2 quick advice today. Well, this session is being recorded and is being translated to Spanish. Our chat box is available for questions and comments. Also, I'd like to send greetings to the people who is joining us through the YouTube channel of Red Clara. Well, we have a really interesting agenda. To start, I would like to give the word to Luis Eliezer Cadenas, the CEO of Red Clara. Please, Luis.
Thank you, Tania. Good morning, good afternoon, and good night to everybody participating in this webinar. I am going to switch to Spanish to take advantage of the translation and to explain to everybody in our language the reason and the impact of this webinar. Bueno, buenos días, buenas tardes, buenas noches a todos. Es un gusto, un placer y me alegra muchísimo la realización de este webinar, de esta actividad con EBRAINS, con quienes hemos venido conversando y trabajando para estructurar una colaboración que permita a Latinoamérica tener, acceder, usar y poder potenciar sus capacidades de investigación en áreas relativas al cerebro, gracias a esta infraestructura y a este proyecto tan importante que originalmente era denominado el proyecto del cerebro humano, The Human Brain Project, que se ha convertido y se está convirtiendo en una organización, una infraestructura de investigación que le presta y le prestará servicios no solamente a los países e investigadores europeos, sino que, como es la costumbre en las inversiones que Europa hace en este ámbito, es una infraestructura abierta que comparte datos, que comparte capacidades, que comparte recursos, para permitir fomentar y potenciar la investigación en la región en esta área de conocimiento. Es muy importante el rol que las redes de investigación y educación juegan en este proceso de articulación. En este caso, el trabajo que hacen nuestras 11 redes nacionales de investigación y educación en cada uno de los países donde están presentes en Latinoamérica, vinculando a las universidades, vinculando a los investigadores. Nosotros a nivel de Red Clara integrando esas capacidades a través de una infraestructura de conectividad, no solamente desde el punto de vista técnico sino también desde el punto de vista humano, y de vinculación de gran capacidad con Europa a partir de el despliegue y puesta en funcionamiento del cable submarino Vela, ¿no? Que nos conecta directamente entre Sines y Fortaleza, y que nos permite pensar en alianzas, en múltiples alianzas de esta naturaleza. La verdad es que el impacto primero de las capacidades de investigación y educación que existen en Europa, al igual que las que existen en Latinoamérica, y la posibilidad de articular colaboraciones para fortalecer mutuamente esos ecosistemas, es un elemento que nosotros tenemos como un objetivo primordial y de gran relevancia para las acciones que hacemos a través de RECLAR. Y por otra parte, el impacto que esto puede tener en apoyar a universidades y centros de investigación de la región a incorporar todas las tecnologías de la transformación digital para acelerar el desarrollo económico y social de nuestra región es un elemento sumamente importante. Nosotros recientemente hemos finalizado la primera fase del proyecto Vela, estamos yendo a una segunda fase donde se está planteando una inversión de alrededor de entre 20 y 30 millones de euros para fortalecer la conectividad de varios países de Centroamérica, del Caribe y otros países como Perú, México, Bolivia, Paraguay, que hasta el momento no cuentan con la misma capacidad de conectividad o infraestructura que hemos logrado desarrollar en la primera fase de este proyecto. Entonces, quiero a todos darles la más cordial bienvenida. Esta es la razón de ser de nuestra infraestructura, la posibilidad de potenciar colaboraciones y cooperación entre todos nuestros actores en Europa y Latinoamérica para fortalecer el desarrollo de todos. Así que bueno, les doy la más cordial bienvenida. Agradezco mucho a EBRAINS todo el trabajo que hemos venido haciendo colaborativamente, y espero que se profundice y se desarrolle a los niveles que lo hemos soñado. Así que muchas gracias a todos y un gusto poder estar acá con ustedes.
Thank you, Luis. And just to continue, I would like to give the word to Steven Bermulin, CEO of EBRAINS and Director of Innovation of the Human Brain Project. Please, Steven, go ahead. You are muted, I'm sorry.
Good morning, good afternoon, and for those in a different time zone, good evening and good night. Allow me 5 minutes to introduce EBRAINS to you, and I'll use a couple of slides for this, if that works. There we go. So EBRAINS is built actually by the Human Brain Project. The Human Brain Project is the big flagship project in Europe around brain research. But EBRAINS is evolving out of that. In the next few years, we will be on our own, and we will build upon the work of the Human Brain Project, so taking it to the next level. And what is EBRAINS doing, actually? I mean, from that scientific work, a research infrastructure was built. That is what EBRAINS is bringing, and it brings so all of the achievements that we've done in the Human Brain Project around brain data and services around that, simulation, modeling tools, quite important in the infrastructure, and access to computing. That is scalable compute, but also supercompute, definitely needed for the simulation tools. So that is in a very, very quick overview what EBRAINS brings. In a bit more detail, what are we building on? So I talked about the Human Brain Project. That's more than 500 scientists and engineers that have worked on this. And actually those results are the ones that are available in EBRAINS. And there were over 200 institutes, academic institutes, some additional international partners that have contributed to this, and more than 1,400 scientific publications. So this is a significant piece of work, significant progress also in the area of brain science that we are delivering to the community and now continuing as EBRAINS. So the ambition is for EBRAINS to become the infrastructure of reference for brain research. What does that mean? It means that we are evolving out of that project and now enabling that to a wide and extensive community of researchers. By the way, that's not only European, that's why we are talking. Secondly, we continue to be enabling the state-of-the-art research, and that is multi-scale research. That is something that specifically comes out of the Human Brain Project, the multi-scale approach towards brain science. That is a whole combination of many different tools, of which we'll have a few introduced in more detail in the coming sessions. But we actually bring all of that together via customizable workflows. Some of them are already there, some can be still built. And that delivers basically a set of unique add-on tools on top of that. There is, for instance, the reinforced inference that allows us to actually do one-shot learning and those kind of additional AI tools that could be used for data analysis, but also for actually model building and model verification. EBRAINS is, like I said, offering this wider range of services, and we've cataloged that into 5 different categories. And with those services in those categories, we want to basically accelerate, as already said in the intro, the brain research. So basically we are enabling the standing on the shoulders of giants in data knowledge, in atlases, in simulation, in brain-inspired technologies. Here you have to think about computers that are based on computer architectures and chip architectures that are based on how the brain actually works, and also in medical data analysis, something that we will further build on. And when I was talking about those giants, well, I've got 3 of them with me, and I'll ask them now to go in a bit more detail. So I will start with Professor Jan Bialy, that will take us through the data solution on EBRAINS. Thank you.
Thank you, Steven, for this big picture of EBRAINS, and I think the perfect introduction for what's coming next. So I'll just give the word to Professor Jan. Professor, just let me, well, you are muted in this moment, and I just want to mention for those joining us that, well, Professor Jan Bialy is professor at the University of Oslo and Infraestructura Director at the Human Brain Project. Now, please go ahead, Professor Jan.
Thank you very much. So hello, everyone. It's my pleasure to present the EBRAINS data and knowledge services. We'll walk through it as systematically as we can in a few minutes, but take first a little historical view on how we have been publishing over the last few centuries. Journals, scientific journals, were invented in the 17th century. Peer review came along in the 20th century. And now in the 21st century, it is for the first time possible to share the underlying research data broadly. This, of course, was something that may have started before, but now it is technically feasible to do it in a digital way. The principles of data sharing can be looked at from different perspectives. If we look at the data and the metadata, we have the FAIR principles, meaning that it is recommended that the data are possible to find, to access, and to work with. Then we have the perspective from data repositories. You need to have trust in the places where the data are stored. And that means that you need to have responsibility and user focus and making sure that the data are stored over a long time and so on. And finally, we have the data governance perspective on sharing of data. That is more like the societal perspective with regard to making sure that there is collective benefit from the sharing of data, that there is control when necessary, and ethics is taking care of these aspects. Now, these FAIR, trust, and care principles are outlined in a few publications, as you can see listed here. Highly cited publications, all of them, because this is, of course, a trend nowadays to go in exactly the direction of these principles. So the importance of access to data is very much related to the need to integrate information. Combining data from different sources, working with complex topics like neuroscience, for example, means that you definitely have to combine information also from many levels of investigation. That means you can answer specific research questions, perform hypothesis-driven research, or data-driven research, where the topic or the question is determined by what you can see in the data. But you cannot have any integration of information unless you can actually find the data, access the data, interpret the data. You need to be able to act on data. And also you cannot do that only based on the publications, which is primarily texts, figures, and some tables, and not really the data. So eBRAINS has a set of services that was just introduced in the previous talk. One of them is the data and knowledge services, which is about sharing and accessing research data, computational models, and software. And a key point here is that these data knowledge services are connected to the other services. So for example, the location of data in the brain can be found through the atlases. The data knowledge service has access to various types of tools for analysis linked to the data that you can find in other services. So here's a picture of what this looks like. So neuroscience covers many, many levels of investigations, from gene, molecule, and through the individual neurons and cell types, networks, whole brain, behavior. And all of these levels represent where the data are. Then you have researchers who provide data and researchers who use data, providers and consumers, and they're all researchers, and they're usually on both sides. So this is the logic. Now, in order to manage the data, not only the publications, in order to manage the data, the research data, we have three pieces in this service. One is a so-called knowledge graph, the other is a metadata model, and the third is a curation workflow, and I'll briefly introduce you to these three elements. So the knowledge graph, what is that? That is the system for managing metadata, information about data about the core data. In the knowledge graph, there is flexibility. Many different entities can be connected. So it means that metadata, for example, describing the data sets, they are linked to other data sets that are similar. They are linked to software tools for analysis and visualization. And all these links exist in such a way that you can keep adding more information. It's dynamic and flexible. And you can actually look at this knowledge graph. It is available online. And here you can see the nodes and the links that exists inside the system. Now, you won't be able to make much use of exactly the graph itself, but that's the basis for delivering what you want in the end in terms of finding and accessing data. But there is not much use of a knowledge graph if you don't have a structure in the information stored inside the graph. So you need metadata that follows certain rules. There is a model for the metadata. We refer to this as OpenMinds, which is our metadata model. And that takes care of standards and conventions. It makes sure that the language is the same and that we don't use the same terms for completely, that we don't use different terms for something that should have the same word. So this applies then to all kinds of studies of neuroscience, and it's gradually being expanded to cover all the different fields. At the moment, we have a lot of things in place. So all the common metadata that's needed for all type of research, they are there. And then we have in-depth metadata for several areas, and we continue to build that into different areas. And it's done in an international collaborative fashion. For example, together with the International Neuroinformatics Coordinating Facility and connecting to groups around the world. But in the end, we need to make sure that information is correctly entered. That is why we have a curation workflow. This is essentially quality assurance in order to make sure that the metadata that goes into the knowledge graph following the OpenMinds metadata model are correctly entered. And this has a number of steps. We're not going to go through the details here, but it starts with checking that the research itself follows certain basic ethical principles and that the regulations are in place for the research, continues with structuring the files so that they can be easily navigated by others. Then the metadata comes in following the OpenMinds metadata model. Then there is a data descriptor that there is some guidance on providing so that you can read about what these data are. licenses is attached to the data, Creative Commons licenses. So thereby you send a message to the user. These are the conditions that apply. For example, data has to be cited. Then they are uploaded in the end to our long-term storage, and they are published online and can be found by others and used by others. So the services are essentially one service for sharing, meaning that that service is addressing the data provider. So if you go to eBRAINS.eu, you will find this service and find out how you can share data and request the curation of the data. That means that the data becomes fair using this definition we had a moment ago. They can be fair at the basic level, at the more advanced level, and it means that, for example, you can meet requirements from journals in terms of sharing, or from funders, which typically nowadays also require sharing. Or of course, if you just basically want to do science in a modern way, you will go along this line and make the data more fair. Then there is the service for finding the data, models and software. That service uses obviously the knowledge graph. And from the knowledge graph, there will be an opportunity to pull out information sorted in so-called data cards. And these data cards then describes the data sets you found and give you basic information. You can also do a query in a programmatic access route, which gives you essentially all metadata. That's also possible. The first one, an online search, is available for everyone. The second one requires that you have an eBRAINS account, which you can get if you request one from your institutional email. Now, this is, of course, part of a landscape where the data can be used. So from the left, data, model, software are curated. There's the metadata around them sorted in a knowledge graph. The data can be connected to an atlas, and they can be used in the context of different tools and workflows. This is the entry point, eBRAINS.eu, where you can find these and other services. And why should we deal with this? What are the reasons? Well, some would say that I have to respond to my funding agency that requests that I share the data. Can I get more out of it? Yes, you can. You will get more citations, more credit, citable DOIs by using this service. And the license will communicate to the community how they can use the data. Or there could be another reason. I would like to make my data available together with a journal publication. Can eBRAINS do that? Yes, we have a special track for exactly that, so that you can actually release your data at the same time as your publication goes online, if you wish to do it in that order. And also, actually, with reviewers having access to the data is a possibility. We have different routes described on the web. Or here, the data from my research is so important or is of a fundamental nature, so I want to make sure that they can be used in the future. Or I'm looking for research data to complement my own research. What should I do? Well, the point is, of course, that you can then go in, deliver data, request curation, have the metadata added to them, and have the long-term storage, using this specialized service also then to find data that can supplement or add to your own research. I like to have a reminder finally that the CV in the future will not only contain publications, it will undoubtedly also contain the data that you have shared. So here's a publication list, and then there is the dataset list. In our system, you can take out the data sets for each researcher. We have 1,800 researchers so far sharing data through our system, and they can pull out the information on how many data sets they have contributed to. And for younger researchers building a career in the future, this is obviously the way to go. This is how you create transparent research, the highest level of integrity and rigorous research. So that's the basic message. And here are some of the teams involved in building this service. And we also like to acknowledge the Phoenix consortium of supercomputer centers that allow us to have the necessary computing capacity and storage capacities for this. Thanks very much.
Thank you, Professor Jan. And at this moment, we have a couple of questions, but I would like to leave them for the end. And so now that we could continue, please, to the presentation about Atlas Services with Timo Dikscheit, professor at Heinrich Heine University, Dulcerdorf and researched, and I hope to pronounce this well, I'm sorry, Forschungszentrum Jülich. Please, Timo,
Thanks a
go
lot.
ahead, please.
Thanks a lot for the kind introduction, and the pronunciation was actually quite good. So you did that very well. Yes, so my name is Timo Dickscheid. I'm working here in Germany, in Jülich, and I'm with many collaborators in the Human Brain Project developing the Brain Atlas services of EBRAINS. And while presenting this, you will see some things picked up from the presentation of Jan Bjali. But before we start out, let me just give you a brief idea what we mean when we talk about brain atlases. So a brain atlas it's of course not a book, but what we usually mean by brain atlas is that there is a reference space, just like we have one of the Earth. That is, of course, a model of the brain. And then in that space, we assume that we have maps which basically color this space. They give color to this space, they give values to this space, partitioning it into certain regions, as we have that for a political map of the Earth. And an important third ingredient is that there is a taxonomy that gives names to these mapped regions, mapped structures, and typically also gives some relationships between them. So for example, here on the right, you see that brain regions have been hierarchically aggregated into a tree of larger structures. So that would be the simple picture of a brain atlas. In many cases, however, as you can have it for the Earth, the situation is more complex because you can map the brain in different ways, just as you could map climate zones and topography of the Earth, in addition to the political structures. You can also, of course, map the brain in different ways. And this is an important aspect of what we do, because we want to provide a multitude of maps of different species and of different principles of brain organization in a well-structured system, building on EBRAINS and building also on the data services that Jan Bjali has just presented. And just to show a few, this is an atlas here of the rat brain that is developed in Oslo in the Institute of Jan Bjali, in fact, which is based on magnetic resonance imaging and delineates the structures in the rodent brain. And you can see here on the bottom how we are developing this further and further in the HBP. So we have now released the 4th version of this atlas with now more than 200 structures mapped. So this is now a fairly complete map of the rodent brain, very detailed, that has been evolved over the years using EBRAINS services. Here in Jülich, we are building an atlas of the human brain, and the centerpiece, so to say, of that is our maps of the cytoarchitecture, where we basically distinguish brain regions according to the arrangement of the cells, as you can see it here on the right, which shows pictures from 2 different brain regions. And this is mapped in different postmortem brains. This has to be done postmortem because we need tissue sections to see this high cellular detail. And we do this in multiple postmortem brains. And these maps are then aggregated into reference spaces to also show the variability across different brains, because in the human brain, this is a very important aspect, the differences in different brains, and to capture this information. So this is an example and the core piece of the human atlas that we have. And we are combining this, these cytoarchitectonic maps also with other maps. So you see here a screenshot of our atlas viewer, which you will see in more detail in a minute. And you see here a part of these cytoarchitectonic maps, but there are also maps that are complementary to this. For example, on this image here, you see maps of long white matter fiber tracks, which shows where we have thick bundles of nerve fibers going for the brain, which is of course a different structure. And also short bundle maps that go from different surface positions to other surface positions in the brain. And there are also parcellations that are built on brain function, which show clusters of functional activity measured in human brains under different cognitive paradigms that patients were doing. These are complementary ways of mapping the brain, of course, that we are integrating into a unique system. And then in addition to this, we try to connect everything to the microscopic scale. And there we have this big brain model, which is an image of a human brain that has been reconstructed from more than 7,000 tissue sections to see the human brain at much larger detail. You can see that here on the right. And this is also combined with the other maps into a system that can be easily explored for users. And of course, at this microscopic scale, we also provide maps of the structures that we are interested in. Now, there is an important aspect in EBRAINS that we want to use, and Jan Bialy has already mentioned that. We want to use brain atlases to organize neuroscience data in anatomical fashion. So to give locations in the brain to data sets, which helps to better understand data sets, but which in turn also helps us to describe the different brain regions in different facets. And here are some, just as a teaser, some nice images. They really look beautiful. This is data from microscopy, from polarized light imaging microscopy that reveals the orientations of nerve fibers in the brain. Beautiful data. These are individual cells of cell bodies. And we are linking such, so we are sharing such data through the EBRAINS data services and linking them with the atlases to arrive at a more complete and more multifaceted description of how the brain is organized. So in total, we have reference brain atlases that give us maps of brain regions at different resolutions and at different species. We have regional features of brain regions, which are themselves rich data sets with a lot of information. And there's a third aspect. We have connectivity data, which describes how different brain regions are connected and which is also in itself collections of data sets. And now this uses the EBRAINS infrastructure. And this slide here shows some commonalities with one of the last slides that Jan Bialy has shown. So we use EBRAINS data curation for all these different forms of neuroscience data to actually put these data sets into EBRAINS, share them through the knowledge graph, and share their rich metadata descriptions in the knowledge graph. So people could already find them there with the tools that Jan Bialy has shown by searching for metadata. The Atlas services are building on top of this. So the Atlas services allow you to browse the brain in 3D, navigate it in 3D, select brain regions, understand the relationships between brain regions, but give you from there access to a whole range of data that is anatomically organized in the EBRAINS knowledge graph. And the EBRAINS Atlas services are a selection of different tools. Most importantly, there are web-based tools that allow you to visually explore the atlases, but there are also programmatic tools that allow developers and data scientists to implement workflows on the basis of brain atlases. And I just want to use the remaining time to show you some of these tools. So this can, of course, all be found on the EBRAINS portal, where you can click and find these Atlas services. And you will see there that this is organized into 3 categories. There are tools for accessing and browsing the atlases, here in blue. Then we have also tools that help people to integrate their atlas to brain atlases, so their data to brain atlases. And these tools are also used by our curation teams. And third, there are also tools that help users to analyze data with brain atlases, so to use brain atlases for data analysis. And I'm just showing examples of each of these 3 categories here to finalize my talk. So for accessing atlases, there is, amongst other tools, the 3D Atlas Viewer. The software is called Zebra Explorer that is available online under the URL that you see here. And it allows you to find all these parcellations and brain models that I've shown, select brain regions, and also find data that is linked to these brain regions, that is characteristic to these brain regions. It also allows us to find such connectivity data sets. Here on screen, we currently see some receptor neurotransmitter distributions that are representative for a selected brain region. And I think this movie is also showing some of the connectivity in a second. Yeah, well, let's just move on. So this is a 3D viewer. Then amongst the software interfaces are also libraries for programmers, and these are for unexperienced entry-level programmers and also for professional software developers, both. In particular, we are relying on the Python programming language. And here you can really use the exact same functionalities, but connect them to computational workflows or even to your third-party software to access atlases
to relate different data sets, retrieve this data, and do data science experiments. This is currently also used a lot for simulation setups in the Human Brain Project to collect experimental constraints to set up simulation models. There are also tools that help users to integrate their data with brain atlases, and that typically means that users have a data set and want to find the spatial location in the brain. So usually this means to spatially anchor a data set to the brain. And here are two prominent examples that we provide on the eBrains portal. One is called QuickKnee, that is developed in Oslo. This allows users to anchor histological sections into the 3D atlas space, and this way project atlas regions onto a 2D section with high resolution information on the brain. And on the right, you see a tool that is called Voluba that helps to integrate 3D volumes or tissue blocks from histological experiments into a brain context. So to do this with a 3D volume. There are also more tools. There are tools to anchor morphologies and geometries to the brain, and so on and so forth. Lastly, we provide tools for analyzing data. And again, here just two examples. There's more. One example is the Quint workflow, how we call it, which involves a tool that is called Elastic that performs image segmentation with machine learning methods. So what users can do with this tool is they can, if you have a 2D image from a brain section, for example, you can use Elastic, you can interactively train a machine learning model to identify certain structures, which could typically be cells, and then anchor this information into the brain atlas space and organize the retrieved features. So for example, the densities of cells by brain regions to get a really, to get derived data that is dependent on brain regions and do your experiments. On the right, you see an interactive tool that we call JUGEX, which allows users to select certain gene expressions in different brain regions and perform a differential analysis whether this gene is significantly has a significantly different expression into brain regions, which allows for many kinds of structural studies of the brain. And with that, I would like to end here. So this was, of course, only a very brief and fast look at the things that we have. Please watch out on the eBrains portal and do not hesitate to get in touch with eBrains support if you have questions and need further help to use our tools. Thank you.
Thank you very much, Timo, for this great presentation. We have had really good information this morning for you. So, well, let's go ahead, and I would like to give the floor to Michele Migliore, that is going to talk about simulation services. He is research director at the Italian National Research Council. Please, Michele.
Thank you. Good morning, good afternoon to everybody. It's a pleasure to give you more information about the modeling and simulation services of the EBRAINS infrastructure. This section of the infrastructure is devoted to anyone at different levels of expertise or background. Anyone that would like to do some computational model of brain circuits, neurons, synapses, whole brain, several different regions, and so on. There are several levels of models that are available to build or to use for anyone without downloading any software, without having any specific background in coding or expertise in computing. In general, we are trying to make it as easy as possible for everybody to do simulations. And so in this slide, you see the architecture of the modeling services of the infrastructure. There are three types of users that we are targeting. Learner, which is basically a student or even a citizen interested in going into more details of what a model or a simulation means in practical terms, or more expert users, or a very expert users in which you can just skip the more easiest part of the graphical user interface and go directly to the simulation engines. Each of these type of users access different types of interfaces, which allow to build a workflow starting from experimental data to the model and to the results of the model, and with different details at the single cell level or the circuit level, using different simulation engines. There are several, not several, but quite a few framework that allow you to do and implement computer simulations in this field, and compare and validate the results doing analysis or validation, and eventually go back and refine the model or use that for different type of other type of analysis or comparison with experimental data. At the bottom of the infrastructure, there are all those behind the scene activities and infrastructure elements which allow you a transparent use of the platform. And of course, everything is connected to the knowledge graph that Jan Bioli has shown you in the previous part of in the first part of this section. And also, this is not here, but of course, we are connected also with the atlases that Timo has discussed before. So the important aspect is that no download, no installation, not even a supercomputing account is required to wet your feet with the complex simulation workflows that are already implemented and installed into EBRAINS. So Timo has shown this picture before. This is one of the web pages of EBRAINS, and we are basically under simulation services. And as you see, there are several different levels of simulations that you can do. There are services basically at cellular level, which means synapses, individual neurons, because of course a lot of it has been shown experimentally that several high-level cognitive functions can depend on the inner workings of the individual neurons. And so we have set up several workflows for implementing single neuron models and simulations. Then there are network level type of simulation in which you are interested in implementing a small circuit or even a large region, but maybe, I don't know, the hippocampus, the cerebellum, or the column of the cortex and so on. But also there are workflows and use cases that help you in modeling based always on the experimental data, whole brain. In that case could be either the rodent brain or even in the very limited case in this at this stage, but it is a very important aspect, a human brain. And also there is a section on data analysis and visualization. I'm going to go into more details a little bit on the interactive workflows for use cases for cellular level simulation, because this is my main field of activity. But on the left side here, you see the different levels and different tools and the workflows that are available on the EBRAINS simulation services. Neuromorphic computing is one of these. The neural robotic platform is also allowing you to simulate intelligent agents with robots, virtual robots, in a realistic environment. And you can put your network made by different types of simulators. And then other types of services. This is a visualization. This is the Arbor together with the Nest and Neuron are the main simulation engines that everybody uses in the field right now. The virtual plane by Victor Gissa and the colleagues in P1 is a way in which you can do in silico experiments for epilepsy-based models. And so let's show a little bit. Okay, so we are now inside the workflows and use cases for the cellular level. And you see here, well, I think that if I can click here and go directly online, we can go here. Okay, so this is an online site. you can select different types of use cases. Workflow trace analysis is the very simplest one. But I will take it as an example of the kind of structure that you can expect from the modeling service platform. You see here there are several use cases that you can use for your purpose by reusing models that are already there, done by Human Brain Project researchers, or even from scientists outside that have included their models on the knowledge graph. You can access it from here. And from this point, from this kind of structure, you can have a feeling of the kind of difficulties and the kind of logical use of the use cases that you can have. For example, in this case, you see that this is a Jupyter notebook for those of you who know already Python, but there are also web applications. This is a very simple point and click interface that allow you to extract electrophysiological features from traces. And you see, it is for everybody. So in contrast with other more complex use cases in which you can, of course, you need to know more about the biophysics or the transmission of the brain circuit that you want to model. And then, so this is not for beginners, but still, by having a very first look, you can have an idea what you can do or you cannot do in terms of what you would like to do instead. Another, if I can go again online, another very useful work case workflow is the in silico experiment. In this case, for example, you can go and play with different brain regions at the single cell level. So we have the somatosensory cortex of the mouse, the cerebellum, the rat hippocampus, or other types of regions. But let me go to the hippocampus. In this case, we have a model of the entire hippocampus of the rat. This is about 500,000 neurons. There are all the neurons that are in the hippocampus of a rat. You can go on in details and do an experiment. You can select, zoom, select, oh, too much. We can select 1 or 2 neurons, and then try to figure out the synaptic, the afferent to a specific neuron, let's say this neuron, for example. And so the circuit will filter the network in such a way to have only the neurons that are connected to that neuron, or we can make, for example, another filter to calculate and having the efferent of this other neuron at this other filter, and you can make the intersection. So basically now you are showing only the microcircuit, which includes these 2 neurons. And then you can go on and decide from which neuron you want to record, from which neuron you want to stimulate, generate a simulation by using different types of synaptic connectivity, properties, activation, and so on. So I think that you can see this is a very useful way to help or instruct people that is interested in doing brain science using a computer. This is a summary of what you can see, you can find in the cellular level workflows on EBRAINS. You can do, this is just a snapshot of the workflow that I showed before. After selecting the neuron, you can do, of course, analysis, raster plot, trace plot, peristimulus time integral, but also different type of analysis. We are also in the process of making it available. This is already out, not linked to the EBRAINS yet, but it will be soon. The Hippocampus Hub or the Cerebellum Hub, these are facility hubs in which you can literally go shopping for pieces that are elements that compose your model. You can go on another website outside EBRAINS, let's say Neuromorpho or ModelDB, cherry picking morphologies, model files, ion channels, traces, and put everything together and build your own model. So I'm sure that you will enjoy this section of the platform. And with this, I think I conclude my presentation. Thank you for your attention.
Thank you, Michele, and for this great inspired presentation. I think you cover different levels with your slides. And now I can call my colleagues. Caroline, now we have in our agenda some words from Stephen that you could please make us this big wrap up or conclusions that we might take, please.
Yes, thank you. As we said in the intro, we've got quite a wide range of services. I hope that you've been able to see how everything is interconnected, and that's really also the power of this platform. It's bringing together very, very advanced tools coming from different areas in science and bringing them together. Actually, that's also one of the key aspects of the platform and of the research infrastructure, is the collaboration over different specialities, because that's where the power comes, and that's where you can have a lot of reuse of what others have done before you. And you can find all of that, as already mentioned by the previous speakers, on ebrains.eu. So you can go there, you can get your account, you can actually use it. It's open for researchers, and you can use it. Now, we also know that if you come to the platform, even though we do these nice explanations, and even though there is trainings and everything available, it is still could be still complex. So how do you get into this? Well, we also have supporting organizations for that. So we can help you with the technicalities. We can help you with the more scientific questions, like how would I do that? There is help available. Depending on your questions, we can route that, and we can help you in building your research on eBrains using the tools that we already have. So most of the time you will find a tool that's doing, let's say, 70–90% of what you need to do. You can adapt it because everything is, as said by Timo, everything is connected in software, so you can adapt it. And that's where the workflows play an important role. That's how you can build your own workflow or use one that already exists. Last point that I definitely want to mention, it was on one of the slides as well. We also have community tools where you can reach out to other researchers in your area, in different areas, and really talk about ideas, new things that you want to do, and see where science is. So that's a very short synopsis of some of the additional things I wanted to mention. Go to the platform, have a look around. You will find many more services than what we've been able to show you here. And it's definitely open, and we welcome you to try out. And that's, thank you for listening.
Thank you, Stephen. And I would like to say that you cover some of our questions that we have already from the people that is joining us from our channels and that is following the transmission. And because the first one, and I have this list for all of you, is that how to contribute from Latin America. As I understand from your words, is that the first step is going around in the website and trying to identify which is the most interesting spot for some researchers on some specific areas of work. Well, the following questions, and I understand you also mentioned some of it, is there any kind of training related, and well, they mentioned this during Professor Jan's presentation, related to data sharing for projects?
Let me answer. Maybe Professor Bialy will want to jump in as well. You're back, Jan. So maybe Jan, you can answer that one.
Yes. So I think the most flexible way of thinking about training is at the moment to go to the INCF training space. Mm-hmm. So INCF.org, that is an organization that captures a lot more than eBrains. But we have a training in material there for the use of data and knowledge services. I would also like to emphasize that if you just basically go into the services, and this I think is a general message, when you go into a service, navigate the eBrains web portal and simply deep dive, you will come to different types of tutorials and key material. This applies across also into what Michele Migliore told you about and what Timo Dickscheid exemplified. We have a lot of material available to guide you. And concretely on data sharing, there is specific instructions precisely how it works that you can find directly from the service pages. Thank you.
Great. I would like to make a link with other questions, and I'm just going to switch the order because I think that this is more related. And we have a question is in Spanish. I'm going to translate it. That says, if it's possible to use material that is available in the educative environment. I can imagine that is questions maybe from someone teaching in some university, asking if they can use it for students and different classes.
We definitely have that use case as well. So it's primarily researching infrastructure, of course, but it is being used for teaching purposes as well, and so we have those capabilities. There are a few examples that we've already done, like for instance, there is we did a training to young students where they could use actually the platform and learn about simulation, learn about how that works, and try it out. So that happens, and we actually can support multiple levels of education on the platform. Of course, I mean, the training courses are not necessarily available. You have to build them, but the tools are available to actually use in a classroom setting as well.
Thank you. Professor, will you add something?
Yes, I could also add, for example, if there is interest in using the research data that you can find through the data knowledge services, that's there's always a license attached to them where you can simply see what the conditions are. So there are a few data sets where there are some restrictions, but that's a very, very small minority. And in most of the cases, you will see exactly that you can simply use it. And if you end up reusing it, the conditions are described. And usually it's only citations, or in some cases, some limitations on commercial use, but no other limitations. So very, very useful for educational purposes.
Thank you. Yes, and I also saw in the last presentation, when Michele mentioned that there are also some parts that are dedicated from not experienced users, that could be maybe students, and they're open so they can also go and take a look around and see how can they engage from really basic phases and stages.
Yes, and if I may, in some use cases, and we are going to do it for all of them, there is also a small icon suggesting and showing that there is the possibility to see an interactive video. So people can just look at the video, interact with the video by answering some questions, and the video will guide them through the entire workflow in a step-by-step way. So those are use cases that can be used very easily by a teacher to show some simple simulation workflow, and then the students can go and run it without any problems, because among the things that are available for students, but also for visitors in general, is a small amount of free supercomputing time. So there is no need to apply for additional accounts on the supercomputing. We can let them run for free. Of course, it is controlled and monitored, but it is also available as a facility to people with no experience.
Thank you, Michele. And I have questions coming to Timo, and they ask about if there are, and I know, Steven, I think you mentioned something a few minutes ago, if there are groups of work open to join current activities around brain atlases.
I don't think Timo is on the call anymore. He had to go.
No.
Yeah.
I could try to fill in.
Yeah.
So we might fill in through giving some examples. So, for example, some of the tools that we have that are connected to the atlasing services, they simply speak to how you can register research data in the Atlas space so that the data fits into the Atlas. These tools are available, and you can see them directly on the atlasing services. I could give an example from RathBrain. There are tools to register data to the Waxholm Space Atlas, which is a common and openly shared atlas. And in that way, you can actually get your data into that Atlas space and make them part of the broader Atlas context. This I would recommend if people are interested, particularly in this route, if they would need some additional help to understand the context, they're most welcome to, well, if you want a ticket to be sure it's followed up, you can simply go to the high-level support team and go to support@ebrains.eu and fill in your ticket so that it's not lost in space, and we will be very motivated to answer your questions.
Thank you. Well, I think that, I mean, from Red Clara, we are now enhancing the platform and trying to make this engagement with our communities and the members of the EBRAINS that are part of Red Clara, because it's really important to bring them all these facilities and trying to make them see how they can engage. I know that some of the participations will now start in basically from the basis, trying to know, getting to know how to bring their work to what it's been done. So I understand that these conversation groups and this way to reach information is really important to us to know how to guide them and how to make them easy to move on in all day, because there's a lot of possibilities, and I think it's really important to make them see them. Stephen, I don't know, I mean, that's all my questions from our side. I don't know if you want to make any more comments or any other message that you would like to bring also.
Well, I think, I mean, I'll just repeat it. Welcome to EBRAINS. That I would say we welcome any researchers. And one thing I think to mention also, so we EBRAINS has evolved out of the big project, the Human Brain Project. When we move on in the next few years, we are welcoming other projects, because that is the experience we want to continue. Research projects working together with the research infrastructure to build the new research infrastructure after that. So that's the future, and we want to continue to support all science, all brain science, and across the globe eventually. Yeah, everybody welcome. Let me leave it there.
Thank you very much. Thank you all for this great webinar, for all the information and your kindness to guide us through all the content. I thank also the people that is joining us in the transmission, and well, I hope to see you soon with more information.
Thank you.
Goodbye.